Okay, so as usual, when you define the coenzymes, so coenzymes generally they are small, they are organics, and they are non-protein molecules that carries a chemical group between the enzymes. So it means that they involve in a redox, for example, NAD and also the FAD. So, okay, so generally they are small, they are organic compound, carbon compound. Okay, they are non-protein molecule, non-amino acids that actually carries the chemical groups. Okay, so it's a cofactor of the enzyme. So generally they are placed under, okay, cofactor. Still remember, we learned about the cofactor. So cofactor can be divided into three groups, right? We have prostatic groups, we have the coenzyme, and also the activator. So coenzyme is this group, okay? So they bind briefly, okay? In this case, I understand they have to bind briefly. Okay, they bind briefly to what? They bind briefly to the enzyme active site, but we don't call them as the substrates. Okay, so what are the coenzymes that involved in the aerobic respirations? So we have this NAD and FAD. So in our syllabus, if you write NAD, FAD short form, no problem. Okay, but what's NAD? NAD actually stands for nicotine amide adenine dinucleotides. Okay, and FAD is stand for flavins, adenines, dinucleotides. Okay, so from the name here, they have the adenine. So it means that they are quite similar to the adenosine triphosphate, the ATP. Can you see that? Okay, because ATP also has adenines. Okay, but difference means that these nucleotides, they are dinucleotides, but they have another group called nicotinamide. This one have the flavin groups. Okay, so both of them actually can be synthesized from vitamins B. Okay, why we take vitamin B? Because vitamin B will be used to synthesize this NAD and FAD. So vitamin, you don't take them actually in a high concentration. So therefore, we have limited concentrations of NAD and FAD. So that's why we have to regenerate them. Can I see that? We have to regenerate them. If no, then without the oxygens, you can't actually continue, right, with the aerobic respiration. And then last one's not required to know the features. Here is the coenzyme A that we're going to discuss about this. Okay, so let us look at these nicotines, amides, adenine dinucleotides. So basically, they are coenzyme found in all living cells. Okay, so we have NAD. NAD is a dinucleotide. So what's mean dinucleotide? Two nucleotides. Can you see that? They have two nucleotides joined together. So consist of two nucleotides joined through their phosphate groups. So one nucleotide contains adenine base, another one contains nicotinamides. Okay, so now we have to understand here, adenines actually also termed as a purine. Okay, now purines means what? Purine actually, they have the two-ring structure. Later I will show you guys. So you can look at the adenine. Okay, sorry, adenosine triphosphate ATP. The nitrogenous base actually consists of two rings. Okay, nicotinamide actually belong to the pyrimidines, only one ring. Okay, so later I will show you what's mean two rings, what is mean one ring. Okay, so the main role of NAD, okay, highlight hydrogen atom carrier. Okay, always remember their hydrogen atom carrier. So in general, if the question asks you what is the role, their hydrogen atom carrier. But if you want to describe more, so we always talk about it, they are the coenzyme. Okay, in which process? In dehydrogenation. Coenzyme in the dehydrogenation process. So second, what they do? Okay, they act as the hydrogen atom carrier. Okay, so carry the hydrogen atom and it itself become a reduced form. Okay, so we do know that NAD exists in two forms, the oxidized form, NAD. So every we put a plus. So none of you guys actually asked me about this, why we put a plus. Plus means oxidized form. Are you clear? In fact, they are charged, positively charged, and reduced form without the charge. Are you clear? I mean the positive charge without positive charge. Okay, so now look at this structure. Okay, look at the structure. So this is one nucleotide with phosphate group. Two phosphate group actually join together. Can you see that phosphate group join together? So this is one nucleotide. This is another nucleotide. So this nucleotide is adenine. It's not, I mean the base is adenine. Okay, so two ring. Can you see that? Two ring. One ring, two ring. So another one is nicotinamides, one ring. Okay, so when they plus the hydrogens, can you see that? Okay, so the reactions here, they will take the hydrogen and also the electrons, okay, and then they will combine. Can you see that from 1H now become 2H? From 1H become 2H. So it's a reduced form. Why? They receive hydrogen atom. Okay, so this one is called as a reduced NAD. Okay, reduced NAD. But do you need to draw the structure? No need. Do you need to know how the reaction takes place? No need. So generally, you know that the reduced form, they have the additional hydrogen atom because they can take the hydrogen atom. So when you take the hydrogen atom, it itself is reduced. Okay. How about flavin adenines dinucleotides? Again, it's a di, so it means it have two nucleotides. So they have the adenines nucleotides and the flavin mononucleotide bridged together through their phosphate groups. Okay, so the main role, again, what is the main role? Hydrogen atom carrier. So if the question asks you for NAD, it can ask you for FAD. Same answer. Are you clear? Both of them, they are same answer. A slight difference if you write more about this, FAD. Okay, remember FAD only in Krebs cycle. Function in Krebs cycle. Okay, function in Krebs cycle for the aerobic respiration, but NAD, both. Okay, in glycolysis, in link reactions, also in Krebs cycle. Okay, so in terms of structures, okay, in terms of structures, you can see that they add actually here, okay, hydrogen and electron. Can you see that? When they add the hydrogen and hydrogen, sorry, the proton and electron is, this is one actually called as a hydrogen atom. So can you see the add one? We can add another one. So that's why called FADH2, but only one comes from the substrates. Are you clear? So FAD, so the reduced form, this is called reduced form. This is reduced FAD. Are you clear? So they can take two. They can take two. Can you see that? One here and one here. They can take two. So this is called a reduced form. Okay, again, write down, guys. So this is the adenine. Do you need to remember the structure? No need. Okay, adenine actually this. So this is a flavin. Okay, flavin. So the entire molecule is termed as the FAD. Okay. So far, okay or not, guys? So what you need to know for coenzyme, just the role. Structure, no need. This is just for your informations only. Okay. Now, coenzyme A. Coenzyme A actually, the functions we need to know, they are acetyl or acetate group carrier. Are you clear? Then they form acetyl coenzyme A. And it's very important to initiate, to bring this acetyl group to the Krebs cycle, so that initiates the Krebs cycle. Okay, so the whole structure, again, can you see that they have this adenine again? Can you see that? They still have this structure called adenine, two-ring structure. Can you see the ribose with the phosphate groups, and then they have a lot. So still remember, we say that acetyl-CoA is not two carbon because acetyl-CoA, coenzyme A, have many carbons. Can you see that? So the two carbon actually refer to who? The two carbon actually refer to the acetyl groups. Okay, so with this, I have done for this coenzyme.